Thank you for joining the webinar, Enhancing Pet Health and Food with Plasma Functional Proteins. In this webinar, you will learn more about spray-dried plasma as a unique ingredient with multiple functions in pet food and its superior nutritional value. Research highlights functional plasma proteins' impressive digestibility and comprehensive amino acid profile, making them the premium choice for pet food formulation. Health benefits. The anti-inflammatory properties of functional plasma proteins have been extensively studied, showing positive effects on gut health and overall immune function. Technological benefits. Functional plasma proteins improve product texture by increasing hardness, enhancing water retention and juiciness, preventing phase separation in wet pet foods, and showing potential for improving the processing of extruded products. Novel uses include gravies and lickables, expanding the pet food applications. Sustainable sourcing. Functional plasma proteins emerge as a sustainable ingredient, transforming the pet food industry's approach to the environmental responsibility. Our presenter today will be Professor Dr. Ricardo Vasconcellos. He has a PhD in veterinary medicine from UNICEF Jaboticabal in Brazil, and postdoc dog and cat nutrition from the same university. He has been an associate professor at the State University of Maringá since 2012. He is the author and co-author of more than 52 published articles and has presented at several pet food science conferences around the world. At the conclusion of the presentation, there will be time for questions. Please feel free to use the chat feature to send questions to Dr. Vasconcellos. Let's start with the discussion on enhancing pet health food with plasma functional proteins.
Hello everyone, it's a pleasure to be here with you to talk about the spray-dried animal plasma in pet food. I'm Ricardo Vasconcellos, professor at the Department of Animal Science from the State University of Maringá. So, about our agenda, as spray-dried is a complex ingredient that contributes for the food processing, nutrition, and presents health benefits. We will explore in our agenda the following aspects. Firstly, related to the market trends for the protein sources, and also we will define what is spray-dried animal plasma. And after, we will explore the technological aspects, application for dry and wet pet diets, and nutritional and physiological properties of these ingredients for pets. And at the final of our presentation, we will discuss some functionalities, especially for gut health and immunity for dogs, cats, and other species that have scientific data published until now. And we will finish our presentation with the Q&A section, and it will be a pleasure to answer any doubt from the audience. So the process developing ingredients involves several steps, but a feasible ingredient for pet food products should meet the following criteria. Firstly, the ingredient needs to be safe, and after palatable, digestible, and if the ingredient also presents functional properties, its inclusion in the diet becomes more feasible, even if it is more expensive than other ingredients. And other point to consider for the pet food development and development of the ingredients is related to the sustainability. And the production of ingredients should comply to welfare standards for animal origin ingredients, and also contributes to a circular economy and minimize the environmental impacts in their production. As protein is a nutrient in evidence, considering the aspects reported here, we will discuss this important protein source that is spray-dried animal plasma. Firstly, protein is an important structural and functional molecule in the body, and it presents many functions like enzymatic role, hormonal role, component from the bones, and also membrane transporter, cell communication, receptor, and component for from actin and myosin in the muscle mass, and also the transporter for many substances in the body. From a nutritional perspective, protein is a source of amino acids for building the body's protein components. And as we can see here in this figure, the dog and cat requirements to compensate the endogenous losses are at least 2 or 3 times greater than those to humans, due probably to their carnivorous metabolism compared to other omnivorous species. Protein sources making up 25 to 50% of the composition of dry food and over than 8% in wet food. Protein sources for kibble diets are typically categorized as conventional or special ones, as we can see here in this figure. And conventional sources are animal or vegetable origin ingredients that meet amino acid requirements, but generally have lower digestibility and cost. An exception, of course, of maize gluten feed that presents high digestibility and low cost compared to other common ingredients, but without any functionality for pets, for physiology in pets. And about the special protein, in contrast, these proteins, in addition to amino acid source, present health or technological properties. These sources tend to have a higher biological value and consequently are more expensive. Some examples of the source are egg powder, spray-dried animal plasma, whey protein, brewer's yeast, albumin, and others like fresh meat, for example. About the definition and processing fluxogram of spray-dried animal plasma, this ingredient is a co-product derived mainly from pork and beef processing for human consumption, playing a key role in the nutritional quality of pet food. And the main steps in the production process are, firstly, the transportation, where the whole blood is transported from the slaughterhouse to the production facility under controlled temperatures and stabilized to minimize risks of contamination. And the second, the processing is started, and during this phase the blood is separated from the red blood cells, and it is then dried using a spray drying process which maintains its water solubility and additional properties of this ingredient. The main characteristics of spray-dried animal plasma are, firstly, the high protein and high essential amino acid content in this ingredient, high water-soluble characteristics that contributes to the production of wet and dry dyes as a binder and also emulsifier. And this ingredient is also rich in functional components like albumin, IgG, and other functional proteins, as we can see here, like acute phase protein, lysozymes, IgA, and complement molecules that contributes for the functionality of these ingredients. All of these components are responsible for the functionality of spray-dried animal plasma. Since wet diets are formulated using a variety of animal co-products, from slaughterhouse. We like, for example, mechanically deboned meat, lungs, kidney, and other co-products from slaughterhouse, rich in protein and also in fat. It is essential to stabilize the product phase to prevent separation after processing. And in this context, spray-dried animal plasma is recognized for its effective role as a binder and also emulsifier, especially for wet diets. As an example, in Brazil, we analyzed the product label from 24 pet food companies, and more than 50% include spray-dried animal plasma in their ingredient lists, probably due to its binder and emulsifier function. And additionally, diets containing plasma are well accepted by the dogs and cats, as we will show in this presentation. As wheat gluten is another important ingredient included in wet diet as a binder, like spray-dried animal plasma, we recently formulated 9 wet diets for cats in pate form, incorporating 0 to 4% of each of their ingredients, and spray-dried animal plasma or wheat gluten. And the ingredient composition of the diets were exactly the same, with spray-dried animal plasma or wheat gluten replacing mechanically deboned meat in the formulation. And we analyzed these foods for texture and also the overall aspects of the foods, and we reported here in the next slide. As we can see here, by the texture analysis, we determined the hardness. Hardness represents the peak of force for broke to the structure. Gumminess was other parameter that we evaluated, that is the hardness multiplied by cohesiveness of the pate, and elasticity, that is the deformation without broke the structure of the pate. And the overall effect of spray-dried animal plasma compared to wheat gluten, regardless of the inclusion level, was nearly twice as high for hardness and gumminess, as we can observe here in the blue bar in the first graph, and also in the orange bar in the second graph. And this demonstrates that spray-dried animal plasma is highly effective in making the pate consistent, preventing phase separation or also deformation. When considering the level of inclusion of the wheat gluten or spray-dried animal plasma, as we can see in this graph in blue bar, the effect of spray-dried animal plasma, in the orange bar, the effect of inclusion of wheat gluten, it was evident significant dose response effect of spray-dried animal plasma related to the increasing of hardness, gumminess, and also elasticity that was observed by the high regression coefficient for the 3 parameters that we evaluated. These results highlight the effectiveness of spray-dried animal plasma in wet diets. Therefore, in the R&D process, it is essential to understand the formulation characteristics to determine the appropriate inclusion level for making the ideal consistency and appearance for the animals and also for the owners in this process, respectively. Spray-dried animal plasma is not recognized as a hydrocolloid like GAMS, but as a binder. It can stabilize products from the phase separation in a similar way. While gums are commonly used in food and pet food products, certain inclusions can affect the digestibility. For instance, it was observed for guar gum that the inclusion at 0.4% in diets for dogs decreased the digestibility and stool quality in the animals. And also for other gums like carrageenan, intestinal inflammation was observed in other species than dogs and cats like rats, mice, guinea pigs, and rabbits. Although spray-dried animal plasma has not yet been studied in dogs and cats, the ability to stabilize product phase suggests that it could potentially replace the effects of gums without any side effects as observed for gums. Regarding the palatability, as previously discussed, this is a crucial factor for dry foods, and two different studies involving spray-dried animal plasma, one published in 2003 by Keeley in dogs and another by our group in cats, showed the very similar results. In the Keeley's study, spray-dried animal plasma was coated at 2% on extruded diet for dogs and compared to the same diet without the inclusion of this ingredient. And in our study in cats, the spray-dried animal plasma was included at 4% in the feed mixture before the extrusion, and the ingredient was extruded together the diet. And despite of the difference of application methods, the results were practically the same, with the foods containing spray-dried animal plasma being consumed 3 times more than the control food, as we can see in the both graphs here. It's important to understand what's the component in the spray-dried animal plasma is responsible for the high acceptance that isn't known until now. About the protein quality of spray-dried has been studied in many species, including humans, and the definition of protein quality, that is the capacity of an ingredient to supply the essential amino acid requirements for the studied species. And protein quality depends on the amino acid profile, digestibility, and also the bioavailability measured by the biological value, or how much the amino acids will be used after the absorption in the digestive tract. Although we do not have enough information about the amino acid requirements for adult dogs and cats, nutritional guides published by organizations like FDF and AAFCO are important resources for nutritionists formulating the commercial diets. When considering the amino acid requirements, both the total amount of amino acids and also the amino acid ratio in a protein source are important. And an unbalanced amino acid formulation can interfere in the metabolism and absorption of these nutrients in dogs, cats, and other species. In this context, amino acid score is used for humans to access how rich or deficient is a protein source in a specific amino acid compared to the recommended amino acid profile in the diet. Amino acid score is calculated by dividing the amount of a specific amino acid, like for example lysine, into the crude protein in the source, divided by by the same amino acid in a reference. And in this case, for dogs and cats, we use the nutritional guidelines for published by FIDIAF and also by AAFCO. An amino acid score lower than 1 indicates that protein source is relatively poor, and that in that amino acid, and while values above 1.0 suggest that the amount of this amino acid is adequate in amount of protein from the diet. Spray-dried animal plasma contains a high amount of crude protein, approximately 8%, and the amino acid score for the main amino acids in dogs and cats are higher compared to the most common protein source used in dry diets, such as poultry byproduct meal, meat and bone meal, egg, spray-dried egg, soybean meal, soy protein concentrate, maize gluten, and wheat gluten. As shown in the figures, the amino acid score for the main limiting amino acids for dogs are the highest, and exception of methionine and cysteine. And phenylalanine and tyrosine are important for cats, but the data are not shown here. And although the amino acid score have not displayed here, they are also high for spray-dried animal plasma. Digestibility is another important parameter in the nutritional evaluation of a protein source, but we currently lack information regarding the digestibility of spray-dried animal plasma for dogs and cats. But Song and co-authors in 2015 assessed the crude protein digestibility of this ingredient for pigs, finding it to be over than 90% as observed for spray-dried egg. And the results for these ingredients for digestibility are higher than those reported for common ingredients or common protein sources like meat and bone meal and poultry byproduct meal that range from 8 to 85% approximately. When the amino acid score is multiplied by the crude protein digestibility of a protein source, it calculates the amino acid score corrected by the protein digestibility, or PDCAs. Compared to the main protein sources used in pet food formulations, spray-dried animal plasma demonstrates the highest score for lysine, as we can see here, tryptophan, and also threonine, an exception of methionine plus cysteine that was lower than other ingredients like spray-dried egg and wheat gluten. Supplementation with DL-methionine in dog diets is commonly recommended, as the most common protein sources are generally have a lower ratio of these amino acids in their crude protein content as observed for other ingredients here, like poultry byproduct meal, meat and bone meal, soybean meal, soy protein concentrate, and others. The choice of a protein source is important, as protein in the diet is relatively abundant nutrient, contributing with a mean of 25% from the total nutrients for dogs. But this ingredient represents a relatively low digestibility compared to starch and lipids. As a result, the excretion of protein-derived components in feces is high. If we consider mean apparent as an example, if we consider mean apparent digestibility of 85% for crude protein in a commercial diet for dogs, protein contributes to more than 30% of the total undigestible substrates available for the fermentation in the large gut. This fermentation produces metabolites such as biogenic amines, ammonia, hydrogen sulfide, and others which are typically undesirable in high amounts in feces because they contribute for the odor of feces and also for the environmental pollution. And therefore, it's important to choose highly digestible protein sources when the goal is to control fecal odor, nitrogen excretion, and prevent dysbiosis in the gut microbiome. Ingredients with high digestibility can play a significant role in this sense. For example, we have a series of total tract crude protein digestibility results here in this graph for various ingredients for dogs determined by the substitution method that we can determine the specific digestibility of the ingredient. The results range from 61% as we can see here for the feather meal to 96% for maize gluten, for example. Although the result seems to be similar for some ingredients, such as fish meal, that is highlighted here in red, and for soybean meal, that's highlighted in red also, that presented 8% of digestibility, crude protein digestibility, and also 84% of crude protein digestibility, with only 5% of variation. There are a significant change in the crude protein excretion in the animals, as we can show in the next slide. When estimating the protein excretion, for example, 1 kilogram of protein from fish meal produce 200 grams of excretion, of crude protein excretion, compared to the 1 kilogram of intake of soybean meal produce 160 grams of crude protein excretion for soybean meal. As dogs and cats require high protein levels in their diets, it's essential to formulate a more balanced and digestible diet instead to high protein. This also helps to mitigate the amount of nitrogen excreted to the environment. And since we lack the digestibility data for spray-dried animal protein in dogs and cats, if we use the data published by Song and co-authors for pigs, that they reported 95% of crude protein digestibility in pigs of this ingredient. The crude protein excretion estimated for spray-dried animal plasma for 1 kilogram of intake from spray-dried animal plasma will be 3 or 4 times lower compared to the soybean meal or fish meal. Although we lack enough information on digestibility of spray-dried animal plasma for dogs as ingredient, a recent study found that incorporating 1 in this blue bar or 2 in the orange bar percent of spray-dried animal plasma into a dog diet through coating increased the crude protein digestibility from 79.57% to 81.46% in the diets. Chile also included 2% of spray-dried animal plasma in a dog diet via coating, as we reported in our study, and observed similar results, decreasing more than 10% in fecal excretion on a dry matter base, as we can see in this graph. In the practical point of view, for the owners, high digestible protein favors lower volume and fecal excretion, and the other for the in the dogs. There are many different types of protein in spray-dried animal plasma, like albumin, that accounts for 50% of the composition of protein in this ingredient. That is probably the highest digestible component in the ingredient. Plasma is also rich in immunoglobulins such as IgG, that corresponds to 25% of the protein, and other functional proteins, as we can see here, like lysozymes and growth factors and acute phase protein, that represents 20% of the protein from this ingredient. These components have been extensively studied due to their numerous biological benefits, in addition to their role as a source of amino acids. The most common health benefits studied in spray-dried animal plasma are the immunomodulation, intestinal health, and neuroprotection. The immunological response, about the immunological response, it's divided in high innate and adaptive response, as we can see here in this figure. And the high innate response is characterized by a non-specific reaction driven by polymorphonuclear cells, macrophages, monocytes, activation of the component system, lysozymes activity, and other reactions, as we can see in this figure. This response is important to destroy pathogens in the gut and also to prevent microbial translocation. Since plasma contains not only immunoglobulins but also proteins with antimicrobial properties and growth factors for tissues, its effects on gut microbiota and immunity have been extensively studied in recent years in many species. Spray-dried animal plasma is typically recognized by the immunomodulatory effects on both high innate and adaptive immune response, and the main actions include, as we can see here, the decrease in activated lymphocytes and neutrophils, reduction in TNF-alpha and IL-1 beta, and also increase in IL-10 and TGF-beta levels in the blood. Direct effects of IgG from the ingredient was also observed, although less than 10% of IgG is recovered after extrusion. It has been found this immunoglobulin has been found in feces of animals, indicating the biological effects too. Those changes in the immune system results in anti-inflammatory properties, microbiome modulation, and also the mucosal integrity. Those changes observed by consumption of spray-dried animal plasma, as observed in many species like pigs, mice, and rats, create a tolerogenic environment in the gut. But these effects have been not studied in dogs and cats. This helps to prevent microbial translocation, reduce the inflammatory stimuli, and are also essential for preventing allergic reaction. And it's important to study this effect in companion animals. The intestinal tract is responsible for digestion, nutrient absorption, as a barrier, while the microbiota present in the gut lumen is responsible for producing nutrients, hormones, peptides, and various metabolites that contributes for the host's health. This balance is essential to prevent diseases. The digestive tract has impressive numbers, like, for example, 100 trillion microorganisms and more than 1,000 species that colonize the gut. And although it constitutes only 6% of the body weight, it's a highly active site, accounting for over 20% of the host's energy expenditure. Additionally, the turnover of this tissue occurs from 3 to 5 days. In the gut microbiome of dogs and cats, 5 main phyla are present, as we can see here. Among these, Firmicutes is the most abundant, as it includes important microorganisms responsible for producing short-chain fatty acids such as propionate, acetate, and butyrate. These fatty acids serve as energy source for both the gut and the immune system, and they help to regulate intestinal pH, which is crucial to prevent the growth of pathogenic microorganisms. Dietary modifications, particularly high protein intake, can alter the composition of the gut microbiome by reducing the lactic bacteria such as bifidobacterium and faecalibacterium, while potentially increasing the pathogenic microorganisms. For this reason, it's important to avoid the excessive intake of protein from the diet, focusing on quality instead the amount of protein in the diets. For cats, the positive effects of prebiotics and fermentable fiber, as we can see in the first table, are evident, as they promote the growth of short-chain fatty acid production, microorganisms such as bifidobacterium, faecalibacterium, and Blautia. However, when cats are submitted to a high protein diet, as we can observe in the second table, the gut environment changes, leading to the development of microorganisms specialized in protein and amino acid fermentation. This shift increases the production of biogenic amines, ammonia, branched chain fatty acids, and phenolic compounds that contributes for dysbiosis and also the increase of odor from feces. Although increased protein concentrations in the diet have been linked to the development of protein fermenting bacteria in the intestine, a recent study found that 8% of spray-dried animal plasma in the diet increased the lactobacillus population, as we can see in these figures, and in microbiome in rats. The authors also evaluated the immunological markers and observed an increased concentration of IL-10 and TGF-beta, which have immunomodulatory effect. And the authors also observed the increasing of the MUC2, that is a protein associated for the mucus production by goblet cells that helps to protect the intestine in the animals supplemented by spray-dried animal plasma. In another interesting study, mice prone to accelerated senescence were fed a diet containing 8% of spray-dried animal plasma compared to a negative control for 6 months. And the IL-6 and TNF-alpha were decreased compared to the control group in the spray-dried animal plasma supplemented diet. And additionally, there was an increasing in the IL-10 concentration, supporting the hypothesis of the immunomodulatory effects of this ingredient, as we can see in these graphs. Previous study evaluated the effect of spray-dried animal plasma not only on immune parameters but also in reproduction, gut health, stress conditions, and cognitive function, but in different species, including some studies in dogs and cats. The aging process in dogs and cats is characterized by several factors, including inflammation, neurodegeneration, sarcopenia, and also the immunosenescence. High-quality protein sources, antioxidants, immunonutrients can help to mitigate the aging effects of on these animals. Spray-dried animal plasma has important characteristics for old animals, like high-quality amino acid composition that helps to prevent the sarcopenia, prebiotic effect by increasing the short-chain fatty acid production and supporting the development of lactic microorganisms and improve the fermentation metabolism in the gut. And immunomodulatory other effect from desirable for this ingredient in old dogs and cats is the immunomodulatory effects by increasing the immunomodulatory cytokines like IL-10 and TGF-1 beta, and decreasing the pro-inflammatory concentration of cytokines like TNF-alpha and IL-6 and other pro-inflammatory cytokines. These properties make spray-dried animal plasma a valuable addition to the diets of aging pets, potentially improving their overall health and well-being. Another issue prevalent in older animals is cognitive dysfunction. This condition is common in senior dogs and exhibits similar pathophysiological changes to Alzheimer's disease in humans. An interesting study published by Su and co-authors reported on the degenerative process occurring in the brains of beagle dogs over 4 years period from 1999 to 2002. And the study observed increase in degenerative areas, particularly as we can see here in the ventricles and limbic region, highlighting the impact of aging on canine cognitive function. These finds support the importance of early intervention and potential dietary extractages to support cognitive health in aging dogs. While there are no studies specifically on dogs, research involving SAMP8 mice prone to accelerated senescence, as we can see here, has provided valuable results. For example, Miro et al. Fed the mice with 8% of spray-dried animal plasma compared to a negative control for 6 months, and the authors evaluated several factors including the memory tasks, cytokines, and oxidative status. About the memory tasks, as we can see in the graphs B and C, both short-term and long-term memory tasks showed significant improvement in the animals supplemented with spray-dried animal plasma after 6 months of consumption from consumption of the diets. Serum cytokine concentration. About the serum cytokine concentration, the study found decreased expression of pro-inflammatory cytokines such as TNF-alpha, IL-6, and NF-kappa beta in spray-dried supplemented group. This change is likely attributed to the immunomodulatory effects of this ingredient, as we can see in the graph from A to Y. About the oxidative status, the authors observed also additional assessments on oxidative status indicated potential benefits in reducing oxidative stress. These finds suggest that spray-dried animal plasma may have a potential to support cognitive function and modulate the inflammatory response, which could be beneficial for aging animals. As we near the end of our presentation, it's important to consider that the choice of a protein source involves various factors, including cost, quality, environmental impact, and increasingly companies are selecting ingredients based on sustainability and safety considerations. In this context, spray-dried animal plasma exemplify a circularity in the economy from the ingredients, as it is produced from the blood, a byproduct from the meat production, and due to its proper treatment, spray-dried animal plasma can be safely used in the diets for both human and animals. As we can see in this heat map, green indicates the better performance, and in red we can see the poorer performance in the column. Despite its high nutritional value, as we can see for the plasma, the high digestibility for this ingredient, this ingredient presents a relatively low impact compared to others ingredients that present high nutritional quality but with high impact, like for example spray-dried egg. And other ingredients like feather meal, for example, they present low digestibility or low nutritional value but with feasible environmental impact. So the spray-dried animal plasma presents good nutritional quality and also a feasible environmental impact compared to others for the acidification, eutrophication, and climate change, as we can see in this table. So, in summary, spray-dried animal plasma is a complete ingredient that offers several benefits. From the nutritional point of view, spray-dried animal plasma is a rich nutritional ingredient that presents one of the highest amino acid scores and digestibility compared to the other protein sources for dogs and cats. From the functionality point of view, it provides prebiotic and immunomodulatory effects as well neuroprotective benefits. And this ingredient presents also low environmental impact, and spray-dried animal plasma meets the high nutritional value and low environmental impacts align with the modern demands for pet food ingredients. So, in conclusion, spray-dried animal plasma is a well-researched ingredient across multiple species and has been widely used in cat and dog diets for decades. I would like to thank you for your attention, and I hope the information provided was helpful, and I'm now available for the Q&A section. Thank you.
Good morning, good afternoon. So my name is Tom Schmidt. I'm the Senior Vice President of Strategic Initiatives here at APC, and so I'm kind of leading the moderated portion of the presentation today. So thank you very much, Ricardo, for the information. Very, very well received. Got a few questions, I think, coming through the chat. I think one of the first ones was that the functional studies were performed using 8% product in the as the inclusion, but then the texture studies with plasma, were only set at 4%. So I think the question is, what would 8% product potentially do in a canned food texture?
Yes.
For?
Okay, Tom, nice to see you here, and it's a pleasure to be here with the audience also. And about the inclusion in canned, in wet diets, 8% is a too high amount because, for example, we tested the inclusion of plasma in a drink for cats, and we use it from 1 to 5%. And with 5%, the drink became solid in the solid phase. For wet diets, when we use 8%, 8% is too high amount. I tested before in our lab from 1 to 4%, and the texture by using 4% is very hard pate presentation. And in my opinion, 1 to 2% is enough to get a good texture for pate form, of course. Okay.
Very good. Thank you. Another question on the sustainability side. The source of the carbon emission data. Could you talk a little bit about that?
Yeah. I performed last year a brief evaluation about the carbon emission by using CIMAPRO software and EcoInvent databases that I have available here. And but of course, I used some inputs and outputs, general, not for the specific company as APC, for example. But as I could see some results, I'm not sure, but if I know APC performed some studies with life cycle assessment. I don't know if in Europe or United States, maybe you can contribute in this aspect. And the carbon footprint is lower than 2 kilogram equivalent per kilogram of products produced. But if I remember, they used consultants to develop this this evaluation by using the environmental footprint method. Okay.
Okay, very good. How about question here on is there a difference in performance between beef and pork plasma?
Yes, pork. Yeah, I'm not sure because I have not enough information about this, and in my I cannot contribute with the audience here, but I can check this information and share with you after check. You usually the studies we have only the information about the spray dried animal plasma produced from pork or beef, and I don't have this information. Okay.
Okay. All right. Another question. Powdered egg is an excellent source of protein for pet food and widely used worldwide. What would be some of the advantages of plasma compared to egg?
Yeah. Yes. Especially when we think egg powder. Egg powder is a high biological value ingredient and digestible, like plasma. And plasma is a high digestible ingredient and also high biological value ingredient also. The difference between them, for example, when we use for wet diets, plasma is a better binder than spray dried eggs. And spray dried eggs as plasma is a good emulsifier also. But for dry diets, when we use plasma, we have data from preference tests for dogs and cats, and plasma is a high palatable ingredient. And about egg powder, we don't have enough data to conclude about the palatability, but I think there are this difference, and other difference is related to the functional properties on health, that spray dried egg is a excellent source from the nutritional point of view, but we don't have any data about the health benefits, for example. Yeah.
Okay. All right. Very good. I have one more question here. So, in thinking about the pet food processing area, what do you think are the main benefits of using plasma in those formulas?
Yes, I think we need to think in different ways, because for wet diets we have different use from dry diets, and for the wet diets, for example, the benefits of plasma is mainly related as emulsifier and mainly as a binder. And this ingredient is well recognized by pet food formulators. And as I mentioned it in my presentation, here in Brazil, we collect the data from some pet food products. And if I remember, 24 wet diets, as I mentioned in my presentation, and 50% of these products used plasma in their composition, and we think this is related to this effect as a binder in the composition of wet diets, and it presents a strong effect as a binder, avoiding the phase separation of a product and improving overall aspects and texture of the foods. And I mentioned in the first question, and it's interesting to see the effect of plasma as a binder, because recently we conducted a test here, a simple test with our students here, and we included from 1 to 5% of spray dried plasma in the formulation of a drink for cats, and concentrations higher than 3%, the drink becomes solid. And we found a better inclusion from 0.5 to 1% in the formulation was enough to get the required viscosity in the cat drink. And for wet diets, as I told you, I think from 1 to 2% is enough to get the effect of plasma as a binder. Okay. And when we think in dry foods, we have low amount of data. I think we need to study more those effects in dry foods. But there are two main functional properties of this ingredient. The first is related to the functional protein, as the plasma is processed by spray drying. It's important. This ingredient is important for the functionality in the process. And recently, we are conducting an experiment here that we are comparing a control diet and a diet with 4% of inclusion of spray-dried animal plasma in the mass of product before extrusion. Mm-hmm. And during the process, we observed the increasing in the pressure in the barrel of sugar. And as consequence, we observed the lower density of the kibble after extrusion and high amount of porosity in this kibble. So it's probably that those effects are important to improve the quality of the kibble. And other aspects related to the dry foods is related to the use of fresh meat. Some pet companies, for example, include high amount of fresh meat in the diets with high concentration of fat. And as plasma present effect as emulsifier, this is important to become the slurry more fluid in the barrel and to improve the quality of the kibble and expansion that is important to make the texture of the kibble. But of course, we need some more data about this, and I think this is important to study.
Okay, very good. Okay. Well, I think from a time standpoint, we probably should kind of wrap things up. We do have a few more questions in the chat feature, but we will follow up with those directly with those questioners here by email. So again, I'd like to thank you very much for your time today, Ricardo. Great information shared. Nice. And thanks for fielding a few of those questions. And I think with that, we will conclude the webinar today. And I thank the attendees for attending as well.
Okay. Thank you, Tom. Thank you. All right. Yes, it's a pleasure to be here.
Have a good day.
Bye-bye.